This video will cover defenses and disease. The learning objectives that we'll cover include describing the pathogen in the movie Contagion and talking a little bit about virus vaccines, explain HIV and AIDS. We'll talk a little bit about immune disease as well as organ rejection, and finally allergy and allergic reactions. So the first learning objective is describe the body's response to the pathogen in this movie Contagion, and also talk a little bit about how vaccination can protect us from viruses. So this movie was kind of cool because it took a very realistic look at the spread of a pandemic virus. So this pandemic virus spread throughout the world, started in Asia, and then due to air travel and different visitors, it spread around Asia, Europe, the United States, and pretty much the world. So again, you might be like, well, that's just Hollywood, but if you've been watching the news the last couple years, there's been the Ebola virus outbreak, Zika virus, some small outbreaks of things called bird flu. So in 2014 and 2015, there was this Ebola outbreak that killed over 10,000 people in Western Africa. And it almost became a pandemic. It almost spread around the world, but luckily it was contained to Africa there. And you might wonder, well, how did this virus in this Hollywood movie come to be? Well, it was actually created by consultants at Columbia University who actually modeled it after a real bat virus. And they also sort of modeled the world response to this made-up virus based on the SARS virus, which had spread a little bit in Asia. So basically it was based more in reality than you might think. So one of the most recent pandemic viruses was this 2009 H1N1 Influenza A that spread around the world. Our seasonal flu is actually a couple different viruses, Influenza A and Influenza B viruses that spread usually in our fall, winter, and a little bit into spring. So one of the hosts of the influenza virus, believe it or not, is birds. And that virus can then spread to humans, pigs, and various animals. So the seasonal flu is actually this Influenza A virus, different versions of it, as well as Influenza B. So some basics for the flu virus, an influenza virus. We basically have subtypes of Influenza A. You might have heard of them, H1N1 or H5N1. These subtypes can then be further broken down into strains. So for example, H1N1 from 2009 might have been different from the H1N1 from the Spanish flu, which struck the world in 1918 and caused the death of, I think, millions of people. So these subtypes or these strains are usually named based on their year and a place. So again, what is the H and what is the N? They're actually referring to these viral particles or antigens. So if you look at an influenza virus, you can see it's got sort of a protein outer shell, a lipid sphere, and then an inner core of genetic material, in this case RNA. So the H actually is, I believe it's a glycoprotein, but for our class we can just think of it as a protein. The H stands for hemagglutinin. The N stands for neuraminidase. Again, these are these proteins on the outer surface of the flu virus. So again, we can imagine that our immune system then can detect these antigens, these viral antigens, and then make antibodies against them. And these are the primary targets of our immune system in the flu virus are the hemagglutinin and the neuraminidase antigens. So we make antibodies against those. An interesting thing, there are 11 versions of neuraminidase and over 18 versions of hemagglutinin, and they can be combined to create the various subtypes of the influenza A virus. So you could have something like an H9N7, and then you can actually have strains of the different subtypes. The other interesting thing about the influenza A virus and the B virus is that the viral genes can change and recombine to create genetic shift and genetic drift. So that will create new subtypes and new strains of each subtype. Now, this creates a problem for our immune system because our immune system is very specific in the antibodies it makes and the response that it makes. So every time you get exposed to a new version of the flu virus, it's as if your body's never been exposed to it before. You don't have any protective antibodies and protective cells ready to defend you. So the constant changes in the flu virus make our immune memory sort of not work at all because we're keep getting exposed to new versions. The other thing you might consider when you thought of the contagion virus or the Ebola virus is, why do some people die and other people survive, right? Matt Damon survived in that. Well, you should probably think of your immune system being different between every person because of our different genetics. You could also consider things like healthcare access and your prior health when you get exposed to the virus would affect your survival as well. But if you think about your immune system, if it's slightly different than the person next to you because of different genetics, that might then make your body respond better or in a better way to make you survive. Again, the survivors of the pandemic would then pass their genes on, and possibly their offspring would have a better survival to that pandemic pathogen as well. How do you stop a pandemic virus? Well, in the movie, they used a vaccine against the MEV1 virus. If you look at our current seasonal flu vaccine, it actually is made to protect you against 2 influenza subtypes, California and Hong Kong, and 1 influenza B virus as well. Now, remember, vaccines are not giving you the virus. What are they giving you? Well, most viruses, at least the influenza vaccines that we use in the shots are going to be antigens from the influenza virus. For example, the hemagglutinin and neuraminidase proteins. So you're not getting the virus. It doesn't even have the viral genes. What you're actually getting is an intramuscular injection of the viral antigens. The virus antigens then trigger your immune system to make protective antibodies. The protective antibodies circulate around, protecting you in case you ever are actually exposed to the flu virus. There are also live attenuated viruses, which are viruses that actually are a full virus particle and they function in your body, but they're usually made to elicit an immune response but not make you sick. However, the nasal live attenuated virus isn't being used this flu season. So do flu vaccines work? Well, according to science, it does reduce the number of flu cases in a year, decreases the number of hospitalizations, and the amount we spend on healthcare. The other thing to remember is that the flu kills, and so vaccination helps protect people. So 250,000 people a year can die due to flu-related illness. In the United States, anywhere from 3,000 to 50,000 in a very bad flu year. And remember, the most vulnerable will be kids and older adults. So again, remember, how does vaccination protect you? Well, looking at cattle that are vaccinated against the rabies virus, the vaccination actually creates protective antibodies that circulate for over 6 months in these cattle. So again, if they're actually bit and exposed to the actual rabies virus, then they should be protected. So our next learning objective is explain how HIV infection disrupts the immune system, leading to this clinical condition called AIDS. So the HIV is a recent pandemic, meaning it spread throughout the world. The spread seemed to appear sometime in the 1970s and 80s, originating in Africa. When it spread to the United States, it's thought that it spread through the Caribbean to New York. Regardless, though, HIV spread throughout the world, has killed millions of people and continues to infect and kill people today. So it is a pandemic. HIV stands for Human Immunodeficiency Virus. This viral infection can lead to a clinical symptom or syndrome called AIDS, Acquired Immunodeficiency Syndrome. If left untreated, HIV infection leads to AIDS and eventually causes death in most everybody. So again, HIV infection, you might wonder, well, how does it spread? You've probably learned before that it can spread through blood contact and blood, as well as secretions and unprotected sex. So I wanted to show you guys the CDC website, and you can see that there are currently 1.2 million people infected with HIV in the United States. About 150,000 of those don't know that they're infected. The typical modes of transmission: male-to-male sexual contact, heterosexual sexual contact, IV drug use, and even in some rare cases from pregnant mothers to the unborn fetus. The other thing I thought that was interesting, if you look at the race and ethnicity of people with HIV, African Americans make up about 50% of people with HIV, but they only make up about 12% of the U.S. population, so they are carrying a fairly large burden of the HIV infection in the United States. So how does HIV infection actually occur? Well, it really starts out as a local viral infection, say in your reproductive tract, and then it spreads throughout the body and then begins to infect your immune cells. And eventually that infection seems strange, right? That a virus that should be defended by our immune system actually destroys your immune system, so that you can end up dying from these opportunistic diseases such as cancer and pneumonia that normally wouldn't kill you because of your immune system. So let's look at an example of how this starts in your reproductive tract. So if we consider the female reproductive tract, if we look at the mucosa lining of the vagina, we see the epithelium and the connective tissue underlying it. If the HIV virus is in the female reproductive tract, it can then infect those epithelial cells lining the vagina and create a local infection. Those epithelial cells, once they're infected with HIV, they'll continue to make the HIV virus. This will create a localized inflammation where our innate defenses will come in to protect us. So things like neutrophils and dendritic cells and macrophages then will come to the scene of the infection to help clean up any destroyed epithelial cells, but then of course to try to defend us. So we see macrophages coming in, dendritic cells coming in to the site of infection. Well, the interesting thing about HIV virus, it likes to infect our immune cells. So it begins infecting the dendritic cells and the macrophages. Also, our lymphocytes are recruited to the site of infection. These T cells then themselves become infected. And so as the HIV virus starts locally here, it begins infecting your immune cells. Immune cells, we know, travel around the body in an effort to protect us and to find other immune cells to activate. When they do this, the immune cells actually spread the virus throughout your body. So the immune cells trying to protect you do the job of the virus and spread the virus around the body. Eventually, the helper T cells, when they become infected with the HIV virus, that's when we get destruction of our immune system. So remember, our CD4 positive or helper T cells are the coordinators of our immune response. And when HIV destroys them after years, that's when you can get this clinical syndrome called AIDS. So it's the loss of your immune system that causes opportunistic diseases such as skin cancer and pneumonia to kill you. So an HIV infection, if we consider when it first starts, it's like any infection, the flu or a bacterial infection in your throat. It causes things like interleukins that might trigger a fever, aches and pains, and so sometimes HIV infection or any infection is described as flu-like symptoms. So when this occurs, the HIV virus will be replicating in your body. The helper T cell levels might be reduced, but eventually you'll reach sort of a steady state where not much is going on clinically with you, but then over the course of years your helper T cell levels will plummet. When those helper T cell levels get too low, there's really no way to fight the virus, and eventually your immune system just doesn't work anymore, and that can cause eventually AIDS and in most people lead to death. Hopefully, though, if you are tested and you know that you have HIV, doctors can treat your HIV infection with drugs and prevent the clinical syndrome of AIDS and keep you healthy and alive for many, many years. But of course, you've got to get tested. So the CDC actually recommends that everyone over the age of, I believe, 13 or 16, everyone up to about 65 years of age get tested at least once for HIV. The HIV virus can be tested a couple ways. One is PCR testing, which can detect small amounts of the virus by testing for viral RNA. Another way to test for HIV infection is to test for antibodies that your own immune system is making. So your B cells will make antibodies against the HIV. The downside of the antibody test is you usually have to wait several weeks after you get the infection before you make enough antibodies to detect. So these two ways can be used to see if you're HIV positive, either the PCR test or the antibody test. An interesting thing about the antibody testing is there's actually a home test that you can buy at places like Amazon and probably even some stores locally. It actually tests for the antibody against HIV in your saliva. And so this OraQuick test can actually test whether you have been exposed to HIV. Okay, our next learning objective is describe the potential cause and consequence of autoimmune disease. So autoimmune disease is basically when your own immune system decides to attack either your cells or your proteins made by cells using things like antibodies and cytotoxic or killer T cells to damage your own body. So you might wonder why the body does that. Well, we'll talk about some possible mechanisms. But first I wanted to show you a list of autoimmune diseases, and it's a very long list, and there's quite a few diseases. You may have heard of things like diabetes or Graves' disease, which involves your thyroid gland. There's also Crohn's disease and colitis, multiple sclerosis, which is when your immune system attacks your nervous system. There's also psoriasis and vitiligo when your immune system attacks your skin. So all of these involve a self-attack, and you might wonder, well, why would my own immune system attack myself? Well, these antibodies can attack your proteins and your cells. The cytotoxic T cells can begin attacking your own cells. One possible mechanism is mistaken identity, and it would be something like this. You could imagine you get exposed perhaps to a bacteria or a virus, and those bacterial or viral antigens then elicit an immune response. Well, if your own markers and your own antigens are somewhat close to the structure of the bacterial antigens, your immune system will then turn and begin fighting against you. So that's one of the possibilities, is that the mistaken identity is causing you to attack yourself. So another theory would be that, in addition to mistaken identity, another theory would be bad training of your T cells. So it turns out that when you're born and you're developing your immune system, actually the immune system cells that detect your self-antigens should either be destroyed through apoptosis or at least be told to be quiet and not attack anything. That's called anergy. And so these self-antigens really should not invoke or cause an immune response, but in some people they do. So perhaps bad training of your T cells. Are autoimmune diseases inherited? There's some link between genetics and autoimmune diseases. Females, for example, have a higher incidence of autoimmune disease. Another possibility is poor activity of T regulatory cells. So we haven't talked about T regulatory cells, but they actually stop or slow your immune system down. And if they're not working properly, again, you could have autoimmune attack. And once you've activated your immune system to attack yourself, those antibodies and those lymphocytes can do a tremendous amount of damage to your tissues. In these examples, the lining of your digestive tract or your esophagus basically destroyed, or in vitiligo, your pigment. In multiple sclerosis, the myelin and white matter of your brain and spinal cord. Okay, our next learning objective is explain tissue matching for an organ transplantation and also describe the mechanisms that would cause rejection of your new organ. So again, what makes our immune system either attack or not attack a transplanted organ? So if we considered a couple people who, one person being the recipient and one person being the donor for a kidney, what would make people a match? So if we consider one person who might have lost their kidneys, or their kidneys aren't functioning and they need a transplant, and let's say you wanted to be the donor, what would determine whether you're a match? So one of the things we have to consider with who is a good match for organ donation, the people have to be a good match in terms of blood type. So let's say the recipient is A positive for their blood type and the donor is O positive. That would be okay. The reason this matters, we're not donating blood, but other cells in the body, such as endothelial cells, make the same blood markers. So in this case, this would be an okay match, A positive and O positive. The other thing that we want to look at are these things called human leukocyte antigens, or human leukocyte markers. These are going to be another set of markers which we haven't learned about yet. They're sometimes called major histocompatibility markers, or MHC markers. We'll just call them HLA. So these HLA markers are another set of markers that are found on the surface of our body cells. Again, we're not just talking about red blood cells now, we're talking about all the tissue and all the organ cells of the body. The interesting thing about the HLA genes, they're located on chromosome number 6, and this region of chromosome number 6 is very different between each individual. So again, if your DNA is variable and your DNA is different, then the HLA markers you actually make, the HLA proteins, will be very different between every single person that you know. So every person in the room would have different HLA markers. The only person that would have the same exact HLA markers as you might be your identical twin. So what do these HLA markers do? The human leukocyte antigens actually help take pieces of proteins and antigens from inside your cells and present them on the surface of your body cells. So in this case, you have your HLA protein with a little tiny piece of protein from inside your cell called a self-antigen, and that HLA and antigen combination then basically marks you as yourself. Of course, your cytotoxic T cells, they'll have receptors that could detect the HLA plus antigen, but you wouldn't want your own killer T cells to attack you, so they don't. Also, you wouldn't want your own antibodies to attack your HLA markers. Basically, this is showing your immune system that you belong in your body. Also, the HLA markers, or HLA proteins, become useful if you're ever infected with a virus, because the HLA marker will hold little pieces of the virus up and present that antigen, so that now your cytotoxic T cells can recognize that HLA antigen combination as being bad. And so in this case, it would detect it as foreign and attack your virus-infected cell, which is good to do because it's just going to make more viruses. Now, if we consider this transplanted kidney, because most people will have slightly different HLA markers, the HLA markers of this transplanted kidney cell, the HLA marker will be slightly different than maybe your own HLA markers. The actual little antigen that it presents might be the same, but because it's the HLA marker combined with the little antigen, that little structure might actually appear different now when your immune system looks at it. So again, that's why these HLA markers are so important. These HLA proteins allow the cell to basically show off little antigens that would normally be found inside the cell. And so this is important for our immune system to know what's going on with our cells. So remember, the little HLA protein and its little antigen will mark you as yourself, but then it'll mark other cells, virus-infected cells or cancer cells, for destruction. So if we go back to our kidney donor and our kidney recipient, they'll have slightly different HLA markers. If we look at your HLA markers and the kidney donor HLA markers, we can see that they're similar, at least two of them, but the third one is different. And so when we talk about matching, they actually try to match these HLA markers, especially with kidney donations, as best they can. So in this case, there'd be 2 out of 3 of the HLAs would match. So of course your immune cells like your own HLA markers and ignore them. And when they look at your donated kidney cell, hopefully they'll ignore them and tolerate them as well. But sometimes those cytotoxic T cells or antibodies can detect those markers and attack them. All right, so again, your HLA markers are going to help your immune system decide who belongs and who doesn't belong. Cancer cells don't belong. Viral infected cells definitely don't belong, and so our immune system can take them out. I thought it was interesting. I checked on the internet. This HLA matching is sometimes done for kidney transplants and bone marrow transplants, but not so much for heart transplants. And I wasn't quite sure why, but I guess for heart transplantation it wasn't as important. They also might test you for antibodies in your blood and in your plasma to make sure you won't attack your new organ as well. So again, think about what happens when you get a transplanted organ. What could the rejection be? It could be your cytotoxic T cells killing your new organ cells and basically rejecting and destroying them. Also, another problem might be something like your antibodies actually destroying them as well. Luckily, you could take drugs which would help suppress your immune system so you won't reject your new organ. So here we're looking at the antibodies attacking our new organ, which can occur too. Also, complement and macrophages and neutrophils all could potentially destroy our new organ. Again, if everything's done right and we take these drugs and we're matched up, hopefully that rejection doesn't take place. Okay, our final learning objective is to describe the mechanisms of inflammation and an allergic reaction. So for example, if you get stung by a bee or you have a food allergy and you get all this inflammation, or even just a regular mosquito bite, what is going on in that situation? So inflammation is really just a non-specific response to something infecting you, or sometimes it's just an injury. So inflammation is this non-specific defense and injury response. Sometimes you get it when you scratch yourself, sometimes you get it when you get a cut. But we'll always have things like swelling and redness and pain, sometimes itching. The goal of inflammation is to help you. It's to get blood flow and leukocytes to the area of infection, and also to isolate the area so things won't spread. You also notice this inflammation when you get swollen lymph nodes or when you get swelling in your skin. So again, it's supposed to defend us, but usually we don't like inflammation, so we take things like cortisone or antihistamines. Allergy is basically another thing that can trigger extreme inflammation, right? When you have an allergic reaction, you're basically getting tons of inflammation, which can cause things like a hive or rash and swelling, watery, itchy eyes, runny nose. What is going on when you have an allergy or a bad inflammation response? Well, you've got these mast cells and basophils, even maybe some eosinophils that release immune chemicals like histamine, leukotrienes, even things that we haven't talked much about called prostaglandins. So you would take things like maybe an antihistamine drug or the newer, more recent drugs, anti-allergy drugs, which block leukotrienes. You also activate maybe your helper T cells, and they start making lots of interleukins, and they don't make your allergy any better, right? They think they're helping out, but they're really just coordinating this immune response and making it worse. Also, your B cells make these special different antibodies called IgE antibodies. IgE antibodies really get mast cells and basophils really ready to respond to the allergic antigens and make your immune response even worse, your allergy even triggered even more. All right, so basically allergy is an inflammation response. It's when your immune system responds to something that's really harmless, but you have a full-blown immune response. So harmless antigens like food antigens or things from animals somehow trigger your immune system to overreact. So this puppy seems harmless enough, and including parts of its skin and fur really shouldn't be any bother to you, but for some reason your immune system reacts to those little proteins or markers. It might start with your B cells producing IgE antibodies. And then those IgE antibodies stick onto your basophils and your mast cells. And those basophils and mast cells sit around in your airways and in your skin. And when you get exposed to that antigen again, they release all kinds of chemicals, histamine, interleukins, leukotrienes, and now you've got inflammation of your eyes, your skin, your airways. So you might have wheezing and difficulty breathing. You might swell up and have a rash. So again, histamine is one of the key chemicals, interleukins, prostaglandins, and so hopefully you take a drug or something that can help block these different pathways and make your allergy not as bad. If you've ever talked or heard about food allergies, the common causes of food allergies, we have milk, eggs, peanuts, the various types of nuts, shellfish, different types of seafood. So again, a food allergy is simply your body reacting again to something that would normally be harmless, right? These proteins in your food are not trying to harm you, but your body responds as if it's something harmful. So again, in this situation, what's going on? Like, why are you reacting to stuff in your food? Well, one of the ideas for allergies, including food allergy and even asthma, is this hygiene hypothesis that basically says that when we're growing up, everything's too clean and everything's too sterile, and our immune system really doesn't have anything to attack, and our immune system learns not to be very tolerant. Our immune system basically reacts to everything. And there's some evidence that suggests that, for example, infants that grow up around a pet, like a dog, have less allergies. And also, this was a sort of a review of all the data on infant diets, and they were showing that the earlier that these infants got exposed to things like peanuts and milk and egg products, the less likely they were to develop a food allergy. So again, it kind of suggests that maybe if you get exposed to more stuff at a very young age, that your immune system then tolerates these things rather than reacts to them. And you might still be wondering, well, I don't still understand, like if you get exposed to a peanut, what makes your body then want to react? And I don't claim to understand exactly how it happens, but for example, through your skin or through your intestines, some people, when they get exposed, and in some ways when you get exposed to the food antigen, your immune system tolerates it and doesn't react to it, so you don't get an allergy. In other cases, for some reason, that food antigen gets past the epithelium and it triggers an immune response as if you're getting attacked by something. Your immune system then gets sensitized to it, and whenever you get re-exposed to that antigen, you have the allergic response. So again, it's really like the immune system in the body reacts poorly when it should tolerate something. Instead, it gets sensitized to it, and whenever you get exposed to that antigen, you get an allergic response. So remember, when you get exposed to anything that causes an allergy, including food allergies, it seems like you get this response where your immune system triggers the B cells to make these IgE antibodies. The IgE antibodies make your basophils, your mast cells, super sensitive to the corresponding antigen. So anytime you then get exposed to that antigen, you get a release of these histamines and prostaglandins, causing local responses like swelling and itching, causing dangerous systemic responses like airway obstruction and an unsafe drop in blood pressure, which we know can be caused by the opening up of all those little arterioles and veins as well. We can also get dangerous arrhythmias, irregular heart rhythms. Of course, you can get these changes in your airways where your throat swells up and it makes it difficult to breathe. In order to counteract all these different changes in your body, one of the things you can use is an EpiPen, which delivers a dose of epinephrine to your body, which hopefully counteracts the drop in blood pressure and opens up your airways as well to help save you. All right, hopefully you're not feeling allergic to college and learning. That's it for defenses and disease. I will see you guys in class.